Hello everyone, welcome to ITLS Academy. Our lecture is Mode of Action of Non-Antibiotic Antibacterial Agents. First of all, we are going to study cell wall. What is cell wall? As we know, shell wall is the outer covering of any cell, bacteria or any other and it also present in plants, and this is the outer covering and it is hard covering of bacteria or plants. This is known as cell wall. Cell wall protects the body and it doesn't allow passes of any molecules into and out of the cell. Exchange of gases is possible. So let's start with cell wall. This structure is the traditional target for a group of antibiotics. From the cell wall, some kind of pores are present. From that pores, gases and some materials exchanged in and out of the cell. So, these are the target for a group of antibiotics. Many of the antibiotics work by degrading the cell wall, which include the penicillin. Penicillin is an antibiotic which work by degrading the cell wall and it inhibits the synthesis of the peptidoglycan, that is a major component of the cell wall of bacteria. Penicillin, but a little noticed report which appeared in 1948 showed that low concentration of disinfectant substances caused cell wall lysis, such that normally turbid suspension of bacteria become clear. Some of the disinfectants are that much highly effective that they can cause lysis of the cell wall even in the minute dose. It was thought that these low concentrations of disinfectant cause enzymes whose normal role is to synthesize the cell wall. These synthesize the cell wall and to reverse their role in some way and effect its disruption or lysis. There are a lot of enzymes available whose a minute concentration can degrade the cell wall and cause the lysis of cell. In the original report, the disinfectants caused lysis of E. coli, that is Escherichia coli, Streptococci and Staphylococci. There are a lot of disinfectants that cause lysis of E. coli, Staphylococci and Streptococci. We can say that here another example is glutaraldehyde, and glutaraldehyde is also owes parts of its mode of action to its ability to react with and provide reversible or irreversible cross-linking in cell wall. As a result, other cell functions are impaired. If there will be no cell wall, cell wall will be degraded, then other functions of the cell will not be function normally, because if there is no boundary, then things that are protected in this particular boundary, that things will not work properly, so the other cell functions are impaired, and ultimately this will cause the death of bacteria. This phenomena is especially found in gram-positive bacterial cells. As we know, gram-positive bacteria have teichoic acid in their cell wall. So, just because of presence of teichoic acid, they have a thicker cell wall than the gram-negative bacteria. So, if there is lysis of the peptide, lysis of this particular teichoic acid or peptidoglycan, then this cell wall will be degraded. When there will be no cell wall, then other functions of the cell that were normally occurring will be stopped, and finally this will cause the death of the bacteria. Cytoplasmic membrane. Cytoplasmic membrane is a membrane found inside the cell wall. Cytoplasmic membrane allow selective process of the things. As we know, the cell wall is not permeable, it only allow exchange of gases, etc., gases and water, etc., but the cytoplasmic membrane is semi permeable in nature. It allows passes of many molecules, micromolecules that are required for the growth and survival of the cell. So cytoplasmic membrane have pores, channels, many other things that allow, even cytoplasmic membrane have receptors. From that point, any ligand can bind and it can function in cell. So, the cytoplasmic membrane is semi-permeable in nature, and it allows passes of many molecules that are required for the survival and reproduction, etc., processes of the cell. Actions on the cytoplasmic membrane may be divided into three categories. The action on the cytoplasmic membrane can be divided in three categories. These are action on membrane potential, action on membrane enzyme, and action on general membrane permeability. If there is any antibiotic that specifically attacks cytoplasmic membrane, then it can function by action on membrane potential. As we know, cytoplasmic membrane have charge differences. This is negatively charged from the inside and positively charged from the outside, just because of the ion differences. As we know that potassium ion are more inside and sodium are outside. So just because of presence of more potassium inside of the cell, there just because of presence of potassium inside, and while sodium and many other hydrogen and potassium also, these are many ions are present outside of the cell cytoplasmic membrane, so they gives a negative charge to the inner side of cell and positive charge on the outer of the cell. If any antibiotic that is targeting cytoplasmic membrane, it can also disrupt the charge distribution, that is also known as membrane potential, and by altering the membrane potential, they can cause the lysis of cytoplasmic membrane. Action on membrane enzymes. Some antibiotics can act on the various enzymes and can disrupt the function of enzymes and cytoplasmic membrane. Some can act on the general membrane permeability. As we know, cytoplasmic membrane allow selective molecules passes of selective molecules as it is selectively permeable. So, if there is any alteration in membrane permeability, then there will be no passage of the molecules that are required for the survival proliferation of the cell. So, this will ultimately cause death of the cell. So, these were few actions of antibiotics on the cytoplasmic membrane. First is action on membrane potential. We have seen an overview on the action of on cytoplasmic membrane, and now we are going to discuss in detail. First is action on membrane potential. We will study how any antibiotic would affect the membrane potential and will cause disruption of the cell. Recent work has shown that bacteria, in common with chloroplast and mitochondria, as we know that chloroplast and mitochondria are also known as semi-autonomous body because they can replicate by their own. They doesn't require any outer machinery, so they are known as semi-autonomous organism. They also act as bacteria because as bacteria is prokaryotic cell, still it duplicates itself. So these are also known as semi autonomous body, and these are function as prokaryotic cell. So chloroplast and mitochondria are able through the membrane bound electron transport chain aerobically. As we know that electron transport chain carried out in mitochondria, or the membrane bound adenosine triphosphate anaerobically. Mitochondria is the powerhouse of the cell and is responsible for generation of ATP. To maintain a gradient of electrical potential and pH such that the interior of the bacterial cell is a negative and alkaline. As we have studied, the interior of the cytoplasmic membrane is negatively charged while the charge on the outer boundary is positive. And this negative charge also cause alkylation just because of presence of OH group. This potential gradient and the electrical equivalent of the pH difference, that is if one pH unit is equal to 58 millivolt, if one pH is equal to 58 millivolt at 37 degrees centigrade, give a potential difference across the membrane of 100 to 180 millivolt. Again I am repeating, if the potential gradient and the electrical equivalence of the pH difference, that is one pH unit is 58 millivolt, and at the temperature 37 degrees centigrade, then there will be a potential difference of approximately 100 to 180 millivolt across the membrane. With the inside negative, this would cause the negative on the inner side of the membrane. The membrane is impermeable to protons whose extrusion creates the potential described. This potential or proton motive force, as we know that mitochondria have a particle that is known as F0 particle, and this is responsible for the ATP generation. ATP generation requires the movement of proton, and there is a proton gradient in the membrane, inside the membrane, and this is known as proton motive force, which work by movement of the proton. This potential, also called proton motive force, in turn drives a number of energy requiring functions, which include the synthesis of ATP. As we know that mitochondria is responsible for the synthesis of ATP, the coupling of oxidative processes to phosphorylation, a metabolic sequence called oxidative phosphorylation, and the transport and concentration in the cell of metabolites such as sugar and amino acids. This, in a few simple words, is the basis of chemiosmotic theory, linking metabolism to energy requiring processes. Certain chemical substances have been known for many years to uncouple oxidation from phosphorylation and to inhibit active transport, and for this reason they are named uncoupling agent. So we can say that uncoupling agents are those agents that inhibit the transport of electrons from one place to another, and that is they cause the reduction in ATP. They are also believed to act by rendering the membrane permeable to proton, hence short circulating the potential gradient or proton motive force. Next is action on membrane enzyme. First is electron transport chain. Hexachlorophene inhibits the electron transport chain. Hexachlorophene is an antibiotic that inhibits the ETC in bacteria and thus will not inhibit all metabolic activities in aerobic bacteria. Next is adenosine triphosphatase. Chlorhexidine has been shown to inhibit the membrane ATPase and could thus inhibit anaerobic process. Chlorhexidines are known for the activity to inhibit ATPase enzymes, and we know if there will not be ATPase, then there will be no conversion of ADP to ADP, and there will be no conversion of ATP to ATP. So we can say there will be no formation of ATP at all. Enzymes with thiol groups: mercury chloride, other mercury containing antibacterials, and silver will inhibit enzymes in the membrane and for that matter in the cytoplasm which contains thiol, yeah, or SH group. Thiol group is also known as SH group and this is a sulfur containing group. A similar action is shown by similar action just like mercury chloride and many others. A similar action is shown by 2-bromo, 2-nitropropane, 1,3-diol or bronopol and isothiazolones. Under appropriate conditions, the toxic action on cell thiol groups may be reversed by addition of extrinsic thiol group or addition of extrinsic thiol compounds, for example cytosine, cysteine or thioglycolic acid. Next is action of general membrane permeability. This is last step of action on cytoplasmic membrane. As we have studied, there were three action on cytoplasmic membrane. First by inhibiting the membrane potential. Second was acting on enzymes, and the third is we are going to study, that is membrane permeability. It means the antibiotic that would act on the permeability on the membrane, and this will affect the passes of many molecules inside and outside of the cell. This lesion was recognized early as being one effect of many disinfectant substances. The membrane, as well as providing a dynamic link between metabolism and transport, serves to maintain the pool of metabolites within it. Treatment of bacterial cells with appropriate concentrations of such substances as cetrimide, chlorhexidine or phenol and hexylrisocrinol causes a leakage of a group of characteristic chemical species. The potassium ion, being a small entity, is the first substance to appear when the cytoplasmic membrane is damaged. Amino acids, purines, pyrimidines, and pentoses are examples of those substances which will leak from treated cells. If the action of the drug is not prolonged or exerted in high concentrations, the damage may be reversible and leakage may only induce bacteriostasis. Bacteriostasis is a stage In bacteriostasis, the cell of bacteria does not grow, it does not proliferate, still, it is a live cell. While in the bactericidal action, this lysis of the bacterial cell occurs. Next point is permeabilization. What is the permeabilization? Drugs able to affect outer membrane integrity have also been exploited as potentiators of antimicrobial agents, that is, biocides. For example, disinfectant, antiseptics, and preservatives, and antibiotics. These are some antimicrobial agents, thereby helping these to penetrate the outer membrane of a gram-negative organism and especially Pseudomonas aeruginosa. Chelators, especially ethylene diamine tetraacetic acid, that is EDTA. EDTA is a main chelator. As we studied, EDTA is the most studied chelator, have been used as potentiator of the action of chloroxylenol. Vara has extensively reviewed the subject of permeabilization, and Iris, Fur, and Russell have described a rapid method of evaluating the permeabilization of Pseudomonas aeruginosa. Next is cytoplasm. Cytoplasm is a content that is present in the bacterial cell. Cytoplasm remains covered by cell wall, and it also covered by cellular membrane, or we can say cytoplasmic membrane. So our cytoplasm is covered by cytoplasmic membrane and cell wall. Within the cytoplasm are a number of important subcellular particles which include the ribosome and oxy and deoxyribonucleic acid. Cytoplasm is a content that contains most of the cellular organelles inside it, and it is surrounded by cytoplasmic membrane and then cell wall, and it contains all of the organelles present. It also contains nucleus, and in the nucleus genetic material is present. This may be DNA, RNA, and anything else. It also contains food granules, it contains mitochondria, and if this is a plant cell wall, then it will contain chloroplast and vacuole also. Enzymes other than those in the membrane are also present in the cytoplasm. There are a lot of enzymes present in this particular cytoplasmic membrane, but some of the enzymes are also present in cytoplasm. Many early studies measured overall enzyme inhibition in bacterial cultures, and a search was made for a peculiarly sensitive enzyme which might be identified as a target interference with which would cause death, and no such enzyme has been found. General coagulations. High concentration of disinfectants, for example, chlorhexidine, phenol, or mercury salts, will coagulate the cytoplasm, and in fact, it was this kind of reaction which gave rise to the epithet general protoplasmic poison. It is already referred to providing an uncritical and dismissive definition of the mode of action of disinfectants. There is little doubt, however, that the disinfectants in use in the 1930s had just this effect when applied at high concentrations. Ribosomes. These organelles, the sites of protein synthesis. As we know, protein synthesis is carried out by the ribosomes. These are responsible for the synthesis of protein, because in the process of translation, when there is a synthesis of protein, ribosomal subunits bind to the mRNA and allow codons to pair with anticodons, and finally the synthesis of proteins occurs. So these are the site of protein synthesis. Ribosomes are well established target for antibiotic action. Both hydrogen peroxide and chloromercury benzoate will dissociate the ribosome into its two constituents. But whether this is a secondary reaction of the two chemicals is difficult to access. If there is any kind of antibiotic that acts by inhibiting the joining of these two subunits, as we know that ribosomes structure, one is a small subunit and one is large subunit, and this is the functional state of ribosome. And if there is any antibiotic that acts on these subunits and in removes ribosome in its constituent, then there will not be synthesis of protein, and this will disrupt the protein synthesis and also the ribosome structure. Although there is no real evidence that the ribosome is a prime target for disinfectant substances. Nucleic acids. Acridine dyes used as antiseptics, that is, proflavin and acriflavin, will react specifically with nucleic acids. Proflavin and acriflavin are antibiotics that acts on the nucleic acid. This may be DNA or this may be RNA. These are antibiotics that act on the genetic material by fitting into the double helical structure of this unique molecule. These antibiotics disrupt the structure and function of the DNA by fitting into the double helical structure of molecule. As we know, there is a lot of gap present between bases of the double helical DNA. So, if these are acrylflavin or proflavin, these molecules can fit inside this DNA and can disrupt the proper functioning of this molecule. In so doing, they interfere with its function and can thereby cause cell death. Next is thiol group. Mention has been made for thiol groups in the cytoplasmic membrane as a target for certain antibacterial compounds. Thiol groups also occur in the cytoplasm, and these groups will also serve as targets. Bronopol, both thiazolones, chlorine and chlorine releasing agents, and hypochlorites and iodine will oxidize or react with thiol group. These antibiotics will show reaction with thiol group. Thiol is sulfur containing group, and this group is known as thiol. Next is amino groups. Formaldehyde, sulfur dioxide and glutaraldehyde react with amino groups. If these groups are essential for metabolic activity, then after the reaction of these compounds with amino group, cell death will follow reaction of this nature, that chlorinated thiazolones as well as acting on thiol group can react with ammonia groups, and after working with amino groups, if these are working in the functioning of cell, then there will be no proper functioning. Highly reactive compounds, that is multi-target reactors. There are one or two chemical sterilants in use whose chemical reactivity is so high that they have a very wide spectrum of cell interactions, and it is difficult to pinpoint the fatal reaction. In fact, it is safe to say that there is no single fatal reaction, but that death results from the accumulated effect of many reactions. One or two specific reactions of compounds in this category have already been referred to. Next is beta propiolactone. This is beta. Beta propiolactone is one example. Beta propiolactone is example of multi-target reactors, or we can say this is example of highly reactive compounds. It will alkylate amino, imino, hydroxyl and carboxyl group. This beta propiolactone will alkylate, that is it will add OH group or hydroxyl group or any other alkylating agent, alkylating amino group, imino, hydroxyl and carboxyl group, and it will change the nature of these groups, all of which occur in proteins and react also with thiol and disulfide groups. As we know that in proteins, amino, immuno and these carboxyl groups are important because these are the natural occurring groups in the amino acid. So if there is any reaction with these groups, then this will disrupt the structure and synthesis of protein. React also with thiol and disulfide groups responsible for the secondary structure of proteins and the activity of some enzymes. These will react with these groups and they will change the nature of these groups, and if there is any change in these groups, there will be change in the protein that is synthesized from these amino acids. So the proper secondary structure of protein will be disrupted and there will be no proper functioning of protein, and also the activity of some enzymes will be affected because as we know most of the enzymes are proteins. So if there is any difference or any abnormality is in these groups, that is amino, imino, hydroxyl and carboxyl groups, then this will affect the activity of many enzymes. We can take one more example, that is ethylene oxide. This is another example of highly reactive compounds or multi-target reactors. Ethylene oxide has a very similar range of chemical activity. Many compounds like sulfur dioxide, sulfides, and bisulfides used as preservatives in fruit juice. When these are used as preservative, and these can also enter the inside the fruit and can alter the properties of the fruit that it actually have. So these are used as preservative in fruit juice, ciders and pears are yet other examples. So these were some examples of highly reactive compounds. Conclusion: the above all that we have studied indicate the range and complexity of the reactions involved in the action of some non-antibiotic antibacterial agents. These were some agents that we have studied. These were non-antibiotic agents, but these were non-antibiotic antibacterial agents. Antibacterial means against bacteria. If bacteria is surviving, they will inhibit the survival of bacteria. So these are known as antibacterial agents because these are against bacteria. The concentration dependent, dual, or even multiple role of many of these substances should be noted. This was the conclusion of the chapter we have studied. So thank you very much guys for watching this session. You can follow ITLS Academy for various courses. You can follow on Instagram, Facebook, Twitter, YouTube, WhatsApp and on LinkedIn, that is a large platform for providing the job. You can also subscribe ITLS Academy on YouTube for getting proper lectures, and you can get the app of ITLS Academy on Google Play Store. Thank you very much again for watching this session.